they have, some of them have regulatory sites or regulatory subunits, and they have catalytic subunits. So they have a polypeptide that contains the active site, and they also contain another polypeptide that binds to that first peptide, and it regulates it. Now, when we say allostetic enzymes, we're talking about enzymes that have a change in their structure when they are bound to the substrate or to a regulator. Now, regulatory molecules are known as allostetic modifiers. They can be activators. They can be inhibitors as well. So you can have negative allostetic regulation, where you have an inhibitor binding to a site other than the active site, and they change the structure of the catalytic subunit. They change the structure of the active site, inhibiting enzyme reaction. You can also have activators, and these activating or these positive allostetic modifiers, they also bind to somewhere else on an enzyme, a regulatory subunit. They change the active site so that the active site can bind to substrates with higher affinity, let's say, or they increase the velocity or rate of reaction, the catalytic efficiency of a reaction. So these modifiers, they can be heterotropic or they can be homotropic. Homotropic is basically when a substrate binds to the active site, or when a substrate may bind to a regulatory site. But usually a substrate binds to the active site and does change the structure of the enzyme, making the enzyme more efficient. These are homotropic modifiers. Heterotropic modifiers are something different. They are different than the substrate. They bind again somewhere else on a site on an enzyme, and they modify enzyme activity. Since they are allostetic, there is cooperativity. And this cooperativity can be negative or it can be positive. Again, okay? So either increasing substrate affinity or increasing the catalytic efficiency of the enzyme, or decreasing either one. So these are the modifiers. Now, these modifiers, these allostetic enzymes follow one of two patterns. They either follow what a system known as a K system, or they follow a system known as the V system. In the K system, what you have here is Vmax is different according to the allostetic regulation. But K50 or K half which is similar to Km in regular enzymes, but K half is the term used for allostetic enzymes. So K half is basically the concentration of a substrate that is needed to have half of Vmax, to get half of Vmax. Now, for those enzymes that follow the K system at different substrate concentrations, and in the presence of allostetic modifiers, whether inhibitors or activators, Vmax will be different, but K half will be the same. Now notice, I want you to notice the allostetic pattern, or the sigmoidal plot, the sigmoidal pattern of the plot. So it looks more like an S. Whenever you see, again, this sigmoidal pattern, you know that this is an allostetic enzyme. Enzyme having different structures. So right here you have, at low substrate concentration, the enzyme rate is low, but then there is a transition phase. There's something happens to the structure of the enzyme where it becomes more efficient at higher substrate concentrations. Now notice that in the presence of an inhibitor, again, Vmax is lower, but K half would be the same. activator, K half would be less, meaning that there is increased affinity, and in the presence of an inhibitor, K half would increase, indicating that affinity is lower, or it gets lower than normal. This is normal. Now, notice something else as well, that is in the presence of an activator, the plot looks more hyperbolic rather than sigmoidal. It still does have a sigmoidal pattern, okay, but it looks closer to being hyperbolic. This is another characteristic of anesthetic enzymes. You can apply the same concepts, by the way, on hemoglobin. Hemoglobin, even though it's not an enzyme, but it's a nice model for anesthetic proteins. Okay? So keep that in mind, because when you study hemoglobin extensively in third year, I think, in the blood module, you will learn about factors that activate or that induce or decrease binding of oxygen to hemoglobin. It's the same exact pattern as you see here. So these are anesthetic enzymes. So now, let's. I'll give you an example of anesthetic enzymes. It's a very famous and well-studied example. It's an enzyme known as aspartate transcarbamylase, or ATPase. Okay, that's the abbreviation. So basically, this enzyme catalyzes what is known as the rate-limiting step. We'll talk about rate-limiting steps later on. But basically, what this enzyme does is it catalyzes the very first reaction in the synthesis of CTP. You'll learn about CTP in molecular biology. It's a nucleotide that is needed to make DNA and RNA. So basically, that's the first reaction. Aspartate plus carbamyl phosphate, resulting in the formation of this product right here, and then you have a series of reactions, a number of reactions resulting at the end in the synthesis of CTP. Okay, so whenever cells need CTP, all they do is that they induce this enzyme, they stimulate this enzyme, it becomes more active. So that's the reaction. So how about the enzyme? Well, the enzyme is really a large subunit, multi-subunit protein. It's made of 12 subunits. Large protein. 6 of these subunits are known as the catalytic subunits, and the other 6 are known as the regulatory subunits. So basically what you have here, is that you have 6 catalytic subunits, 2 trimers, that is, they exist in the middle, and then you have 3 dimers, that is the regulatory subunits. Since it is an allosteric protein, it follows, or it has 2 structures, known as the T and R. Again, same terminology as that used in hemoglobin. So you have T and R states, or you have T and R structures. The T state is the less active state. The R state is the more active state. Okay? So that's the enzyme. So let's talk about regulation of this enzyme. So again, there is, whenever CTP is needed, the enzyme switches from the T state to the R state. It becomes more active. Okay. Now the thing is now, how about regulation? So it follows, again, a sigmoidal plot, as you can see here. Now, the regulation of this enzyme takes place by 2 molecules. One of them is the final product itself, which is CTP. In the presence of CTP, what happens here is that you see the shift to the right. So the enzyme becomes less active. It requires, Vmax is the same, but Kn would be different. Actually, in fact, it follows the the same thing. But basically what happens here, this is a problem with textbooks actually. There's a lot of contradiction in these textbooks. But the idea here is that what happens is that if there is large amount of CTP, it means that there is no need to make more of this, more of CTP itself. So if you go back to that reaction, the final product is CTP. What CTP does is that it comes back and it binds to that enzyme, the ATK's enzyme. It binds to the regulatory sites. Okay? So what happens here is that CTP will bind to the regulatory site, stabilizing the T state of that enzyme, making it less active. Okay. So it decreases binding affinity for the substrate enzyme, for aspartate. So, okay. Now, another regulator is ATP. Now, CTP is a pyrimidine, right? So, it's a green, you remember that? CTP is a pyrimidine. So, there should be a balance between pyrimidines and purines. There should always be a balance between these two. So again, when CTP level goes up, it means that its synthesis must stop, must slow down. So it binds to the enzyme and it decreases its efficiency. Now, on the other hand, if CTP level goes up and ATP level if CTP level goes down and ATP level goes up, that you have more purines. This means that this enzyme must be activated to produce more CTP. Again, to balance between purines and pyrimidines in the cell. So you have ATP actually as an allostatic modifier. In this case right here, notice that you have a shift to the left whereby the enzyme becomes more active. That is, it binds to more substrate at lower concentration of that substrate. And it becomes more hyperbolic. Okay. Now, notice something that CTP is an inhibitor that results in changing that it changes the activity, again, the activity of the enzyme, Tmax of that enzyme. Okay? So you have these, you have the inhibitor, CTP, and you have the activator, ATP. Okay. بس على الصوت. والله يا ريت. مين يا شباب؟ ثلاثة لغات. So we'll continue talking about regulation of enzymes by changing their structures. So we finished talking about allostatic regulation. We'll talk about regulation via modulators, that can be activators or inhibitors again. Now, let's review this. Let's review this pathway. Again, you don't have to memorize it. But this is something that you will cover next semester in the metabolism course, biochemistry metabolism. Okay, so basically what happens here is that metabolism of glucose is regulated hormonally. So whenever you need glucose to increase in blood, you have the release of two enzymes, like these are epinephrine and glucagon. Whenever you have high glucose in blood, the pancreas secretes insulin, and insulin activates the pathways that result in storing glucose in liver. Whenever you're fasting, whenever you need glucose in blood so that cells can utilize it, it needs to be released from glycogen that is stored in liver. So you have these two enzymes, epinephrine and glucagon. Now, glucagon, they do the exact same thing, but on different tissues. Glucagon acts on liver cells, stimulating liver cells to release glucose from liver, from glycogen of liver. Okay? Epinephrine, or adrenaline, is the hormone that acts on muscle tissues. Muscle tissues have some glycogen. And what epinephrine does is that it induces muscle cells to release glucose that is stored in glycogen. Okay? So what happens here is that epinephrine or glucagon binds to a receptor on the cell surface, inducing a signal, transmitting a signal. Eventually what happens is that you have an increase in cyclic AMP. This is a modifier, a modulator. Cyclic AMP binds to an enzyme known as protein kinase A. Remember this name. It's really a really important enzyme for metabolism. So what protein kinase A does is that it phosphorylates an enzyme known as glycogen synthase, inhibiting it. And what it does is that it phosphorylates an enzyme known as phosphorylase kinase, activating it. Okay? And then this phosphorylase kinase, it's a kinase. What it does is that it phosphorylates an enzyme known as glycogen phosphorylase, activating it. Now, here what you have when this is inactivated, glycogen synthase, what it does is that it inhibits glycogen synthesis. When the phosphorylase is phosphorylated and it gets activated, inducing degradation of glycogen. So this is really a very nice and efficient and smart system. By one hit, just activating protein kinase A, you have inhibition of glycogen synthesis, elongation of glycogen, and you have stimulation of glycogen degradation. Just one molecule does that. Now notice something. So we'll talk about protein kinase A first before talking about the other enzymes or the effect of phosphorylation. So again, so you have, this is a master enzyme. Protein kinase A, when it's activated by binding to cyclic AMP, it phosphorylates many enzymes and proteins, including these. Again, this becomes inactivated, and this becomes activated by protein kinase A. So this glycogen synthesis stops, and glycogen degradation becomes activated, okay, releasing more glucose from glycogen in muscles by epinephrine and in liver by glucagon. So what is cyclic AMP? It's a small molecule. It's a secondary messenger, as we call it. It's a small molecule that has really large effects. Okay? So, cyclic AMP is a secondary messenger, a small secondary messenger. It's synthesized by an enzyme known as adenylyl cyclase or adenylate cyclase. When this enzyme is activated, it converts ATP to cyclic AMP. Okay? Now, cells must terminate signaling inside them. So what they do is that they reduce the level of cyclic AMP. Eventually, they reduce the level of cyclic AMP by another enzyme known as phosphodiesterase, okay? Converting it to AMP. Okay. So what happens actually is that when these are when these are released, and they bind to the receptor on cell surface, you have activation of adenylate cyclase, the enzyme that synthesizes cyclic AMP from ATP. cyclic AMP, what it does is that it binds to protein kinase A. Protein kinase A is a tetramer. It's a large molecule. It's made of 2 catalytic subunits and 2 regulatory subunits. Whenever it's inactive, in the inactive state, it's formed as this large complex. It has this large structure. The 2 regulatory subunits bind to the catalytic subunits, inhibiting them. Okay? Inhibiting these catalytic subunits. Now, what happens is that, again, protein kinase A, when it gets activated, it phosphorylates many proteins. Now, the thing is, when cyclic AMP level increases, it binds to the regulatory subunits, releasing the 2 catalytic subunits. Now they are free. Now they can phosphorylate many proteins, including those 2 that I told you about, the phosphorylase kinase and the glycogen synthase. Okay? So here we have really big change in the structure of protein kinase A. It loses its quaternary structure, and that's an important point. Now, it does have a quaternary structure because it has multiple subunits that form this enzyme. Now, what cyclic AMP does is that it binds to these regulatory subunits, releasing them. So each polypeptide becomes by itself, free. So there's no quaternary structure anymore. So this is an example. Now, what happens there, or in another mechanism of regulation of enzymes is known as by covalently modifying enzymes. One of the most famous mechanisms of regulating enzymes is a process known as phosphorylation. That is addition of a phosphate group to enzymes. And these phosphate groups are added, I don't know what they are, histidine, but usually it's these 3 amino acids that get phosphorylated. Tyrosine, serine, and threonine. Forget about histidine. These 3 amino acids get phosphorylated, resulting in affecting or modulating the activity of enzymes. Now, and I said modulating the activity of enzymes regardless of whether it is inhibition or activation, because phosphorylation may result in protein activation or it can result in protein inhibition, just like the example that I gave you first, right here. So here, in this example right here, protein kinase A phosphorylates this enzyme, inhibiting it, and phosphorylates this enzyme, activating it. So the effect of adding a phosphate group to enzyme is dependent on the enzyme itself. Okay. Now, enzymes that are responsible for phosphorylating proteins are known as kinases, and they belong to the transferase class of enzymes. Now, these enzymes that get modified by phosphorylation must be, again, regulated by removing the phosphate group. And the enzymes that remove the phosphate group are known as phosphatases. مين يحكي لي الphosphatases to which class of enzymes do they belong? You're right. Hydrolases. They use water to remove phosphate groups. Okay? So, question is, why is phosphorylation really an effective mode of regulation? Because it's really fast. It takes few seconds, if not less than that, to activate or to modify an enzyme. And this modification has huge effect on the enzyme structure because it has multiple negative charges, and these negative charges will change the structure of the enzyme. So it's really a fast mechanism of regulation. The other thing is that Which means what? It means that this protein kinase A can modulate multiple kinases, multiple enzymes by phosphorylation. And these kinases themselves can modify other proteins and other enzymes. So what we have here is an amplification effect. Let's say, for example, that you have, let's go back here to this slide. So let's say that you have 10 receptors on the cell surface, usually it's more than that. Let's say that you have 10 receptors. These 10 receptors would activate 10 adenine cyclases. These 10 adenine cyclases would produce a lot of cyclic AMP, let's say 1,000. Now, these 1,000 cyclic AMPs would activate also large number of protein kinases. Protein kinase A would phosphorylate many of these phosphorylase kinases. And each one of these would activate many of these phosphorylase enzymes. So you're talking about protein kinase A, multiply that by, let's say, 100, and each one of these 100 would activate or modify another 100. So you're talking about 100 times 100, you're talking about 10,000 molecules that can be phosphorylated all at the end. So this is the amplification effect. تفضل.
لما الريجولاتوري سبيريتس
لما
الريجولاتوري سبيريتس يطلق فيهم السايكليك اي ام بي يتصلوا عن بعض ولا لا؟
Yes. البروتين كاينز A. Well, actually, I'm not sure. Yeah, it's shown in the figure here that they are still in contact with each other. So maybe they are.
يعني في quaternary structure.
يعني في quaternary structure. في quaternary structure بس هذول بس الكتاليتي سابينس do not have this quaternary structure anymore. So phosphorylation is really an efficient way of regulating enzymes because of these reasons. Now there are other types of modifications of enzymes, including adenylation, that is addition of AMP to enzymes. Now AMP is a large modifier. It changes greatly the structure of proteins. Also you have uridylation, that is the addition of, again, you have the addition of AMP to molecules as well, to enzymes. And you have other modifications like what is known as ADP ribosylation, the formation or the addition of NAD to enzymes, again, really large structure. Acetylation, the addition of acetyl group. We talked about acetylation of histones as an example of modification of proteins last semester. Okay, so here we have a phosphorylation cascade, whereby protein kinase A phosphorylates multiple enzymes regulating glycogen synthesis or degradation ultimately. So this is an excellent example. Questions? So let's talk about glycogen phosphorylase, which is this enzyme right here that functions in stimulating glycogen degradation. This glycogen phosphorylase is an enzyme that uses a phosphate group in order to release glucose from glycogen. So this phosphate group gets attached to glucose. So when glucose is removed from glycogen, it's removed in the form of glucose 1-phosphate. Again, these details you don't have to know them, but they are important for next semester. So glycogen phosphorylase is a really interesting enzyme because it actually exists in 4 forms, not only 2 but 4 forms. These 2 forms are the following. You have what is known as a T state, and they also have an R state as well. Okay? So there are 2 forms. You have a T state and you have an R state. The T state, again, same concept as any other allostatic proteins or enzyme, is the inactive form of the enzyme. The R state is the active form of the enzyme. Now, these 2 states, or this enzyme, also exists in 2 other forms. So the first form is T or R. changing a T state protein into an R state. And the second modification is phosphorylation. When the enzyme is not phosphorylated, it's known as phosphorylase B. When the enzyme is phosphorylated, it's known as a phosphorylase A. Okay? So again, the enzyme exists in 4 forms. And they are named phosphorylase A or B. Phosphorylase A is the phosphorylated form. Phosphorylase B is the dephosphorylated form. And the other 2 forms are structural, T state, and R state. Now the thing is, changing a T state from an R to T or T to R is also affected by phosphorylation state. That is, when it is dephosphorylated, when it is phosphorylase B, it exists mainly in the T state. And when it is phosphorylated, it exists mainly in the R state. Okay? So these 2, R state and T state, they exist in equilibrium. Whenever the enzyme is dephosphorylated, the equilibrium shifts towards having it more in the T state. And when it is phosphorylated, the equilibrium shifts to the R state. Okay? So again, we have 2 types of modifications. So this is the effect of phosphorylation, having it's either phosphorylase A or phosphorylase B. Now, what's important about this slide or this piece of information is that what's phosphorylated is a serine, and serine exists away from where the active site is of this enzyme. Yes. Okay. So glycogen phosphorylase exists in 4 forms. Okay? 1, 2, 3, 4. The first 2 forms are structural. Okay? The enzyme either exists in a T state or an R state. Same concept as hemoglobin and other proteins, allosteric proteins. Now, in the R state, the protein is more active than in the T state. Okay? So these are the 2 forms. Now, the other 2 forms are related to phosphorylation. If it is phosphorylated, it's known as phosphorylase A. If it is not phosphorylated, it is known as phosphorylase B. Okay? So we have 2 states again, R and T, and the enzyme is either phosphorylated or not phosphorylated. Now, how are they connected to each other? How does phosphorylation affect the structural state of the enzyme? Now, if it is not phosphorylated, usually this is R state, this is the T state. It exists in equilibrium. Okay? When it is not phosphorylated, the equilibrium shifts more towards the T state. So what phosphorylation does is this right here. When it is phosphorylated, the equilibrium shifts to the R state. So what happens here is that what phosphorylation does is that it makes it, there's more tendency, it makes the enzyme likely to exist in the R state. Okay? But not necessarily. So in the 4 forms, related to the phosphorylation, covalent modification, or structural modification, R and T. Now, when it comes to phosphorylation, when it's phosphorylated, it's known as phosphorylase A. When it's not phosphorylated, it is known as phosphorylase B. Now, the thing is, the other thing is that the switch from T and R is related to how much energy the cell has. So it's related to ATP and AMP. There is a balance between ATP and AMP. And you will learn happens here is that whenever you have high level of ATP or high level of glucose 6-phosphate, the protein becomes more in the T state. ليش? So not to waste energy and not to waste glucose. Then glucose, what does phosphorylase do? It releases glucose from glycogen. Whenever energy is needed. So whenever ATP is high, it means that cells have sufficient energy. So there is no need to release glucose from glycogen. Let's keep it as is. So ATP binds to the enzyme, to the phosphorylase enzyme, especially in the dephosphorylated state, and it stabilizes the T state, inactivating the enzyme. Whenever AMP is high, meaning that energy is lacking, energy is low in these cells, AMP binds to the enzyme and it stabilizes the enzyme in the T state. So we have two ways of regulating the structure of this enzyme. One, by phosphorylation. So phosphorylation shifts the enzyme from the B to A. Whenever it is in the A, it directly goes into the active state, the R state. That's one. The other thing is structural, that is TNR, or regulation by energy. If there is high level of energy, what happens is the ATP or glucose 6-phosphate bind to the enzyme, stabilizing the T state. Okay? And the opposite is true with AMP. Whenever energy is lacking from cells, AMP binds to the T state and it switches the enzyme into the R state. So there are two modes of regulation. So this is exactly what I said in terms of regulation. The same thing. So for phosphorylase B, we have two modes of regulation, either by phosphorylation or by the energy charge. Again, same concept here. It's all in these slides. Any questions? Okay. So we can also regulate enzymes differently as well. And here what we have is regulation of regulation by large regulatory molecules. In other words, previously what I said is that we can modify enzymes like protein kinase A by cyclic AMP. Cyclic AMP is a small molecule. But we can also regulate enzymes by large molecules or large proteins. A great example is G proteins. There are two G proteins in cell biology. So these G proteins, again, these are trimers made of 3 subunits, alpha, beta, and gamma. Alpha is the active subunit. Again, it's not an enzyme. Beta and gamma are regulatory subunits. Now, G protein is bound to a 7-transmembrane domain receptor, known as G protein coupled receptors, GPCRs. A ligand, like an agonist, binds to this receptor. The structure of the receptor changes. It makes G protein able to bind to that receptor. When G protein binds to the receptor, there is a change in the structure of G protein. The alpha subunit is released. It gets released. When it gets released, well, what happens actually, GDP is bound to the alpha subunit. Whenever the G protein binds to the receptor, GDP is released, and we have entry of GTP. When GTP binds to the alpha subunit gets released. Now, this alpha subunit then can bind to effectors, can bind to other proteins, including enzymes. So, when I said here in this example right here, epinephrine and glucagon, and I said, well, what you have is a hormone. It binds to the receptor. The receptor is actually a 7-transmembrane domain receptor. That is GPCR, G protein coupled receptor. It changes the structure. It can interact. It activates a G protein. And the G alpha subunit binds to adenylate cyclase, and adenylate cyclase increases the level of cyclic AMP, activating protein kinase A, you have this phosphorylation, and so on. Now, you also have, so the G proteins are known as large G proteins because they are large. They are made of 3 subunits, alpha, beta, and gamma. You also have what is known as monomeric G proteins because they are not trimers. They are small molecules, but they are also regulated by GTP and GDP, just like G proteins or the G alpha proteins. So what you have here is a protein that is bound to GDP, and you have replacement of GDP. GDP is released, and GTP binds to it. Now this is the active form of the protein. Again, these are not enzymes. Now, and it's a cycle. It's a cycle between what is known as exchange and hydrolysis. So here, what you have here is exchanging GDP by GTP, and it's regulated by these proteins, which are known as guanine nucleotide exchange factors. What these do is that they bind to monomeric G proteins, and they release GDP from these proteins, allowing for the triphosphate to bind to these monomeric G proteins. Now, to inactivate this protein, you have to activate hydrolysis by proteins known as GTPase activating proteins that hydrolyze the triphosphate to the diphosphate. Now, what's really important as well is that the GTPase, so what GTPase, it's an enzymatic activity. It's basically an enzymatic activity that converts the triphosphate to diphosphate. And this enzymatic activity is intrinsic. Intrinsic. ايش يعني؟ يعني انه it's internal. It's a characteristic of the protein itself. It has an enzymatic activity. So these monomeric proteins, G proteins, have enzymatic activity, even though they're not enzymes, because this enzymatic activity affects the protein itself. It does not affect other proteins. Same thing with the G alpha. Same thing with the G alpha right here. The alpha subunit also has a GTPase activity that is intrinsic. So what it does is that GTP is hydrolyzed by the protein itself, converting it to the inactive form. But the enzymatic activity of G proteins, whether trimeric or monomeric, is regulated by proteins like GAPs, or GTPase activating proteins, or GEFs, guanine nucleotide exchange factors, here, or by GDIs or GDP dissociation inhibitors. What they do is that they bind to the monomeric G protein or the trimeric G protein, and they prevent the release of GDP from the protein, so it stays inactive in the inactive form. So we have numbers 1 and 3 are inhibitors of G proteins. Number 2 is actually an activator. Yes. شو؟ هذول مين؟ exchange factors. هم proteins لحال. يعني they are independent proteins. The cytosol, and what they do is they bind to the G proteins, whether monomeric or trimeric. لما يربطوا معهم بيعملوا stimulation, بيغيروا الستركشر again. It's all about structure. بيغيروا الستركشر تبع البروتين بيخلوا سهل انه تطلع الجي دي بي، او سهل انها تطلع تفلت من البروتين، او بيخلوها صعب زي هذول الجي دي ايز، او انه زي الجاز انه بيحفزوا الجي دي بي اس اكتيفيتي اللي جوا البروتين اللي بتعمل انكتيفيشن للبروتين. يس. receptor targeting kinases. انه فيه كاينيز اكتيفيتي بيعمل
بروتين مش
بس هو بروتين ريسبتور. هذا ريسبتور تايروسين كاينيسز ممكن يشتغلوا على حالهم، يعني بيعملوا اوتو فوسفوريليشن، واي ثينك انه في منهم بيقدروا انه يعملوا فوسفوريليشن لاذر بروتينز.
اول هيك بيعمل
بيعملوا اوتو فوسفوريليشن بس ممكن يعملوا فوسفوريليشن لاذر بروتينز. بس لاحظي شغله، انتبهي شغله على اوتو فوسفوريليشن. انت عندك الريسبتور بكون مونوميريك، يعني كل شين لحاله. لما الليجند يربط الاثنين بيربطوا على بعض، هذا بيعمل فوسفوريليشن لهذا وهذا بيعمل فوسفوريليشن لهذا، مش فبسموه اوتو فوسفوريليشن، بس مش انه نفس الشين الباركتايد بتعمل فوسفوريليشن لحالها لإلها. فهو كاينه، هو actually it is an enzyme لانه عنده substrate. السبستريت هو شين زي اخوه. اوكي فكل الاعمال اللي نحكي عنها احنا so far الان هو changing enzyme activity by changing the structure of enzymes. فحكينا على الالوستيريك بروتينز كيف الستراكشر تبعهم بتغير، بصير cooperative sigmoidal whatever. حكينا عن التشينج باي فوسفوريليشن وكيف انه الفوسفيت جروب بتغير برضه الستراكشر تبع الانزيم. وحكينا على الجي بروتينز كيف انه actually what they do انه هذول large proteins what they do انه they bind to enzymes مثل adenylate cyclase وبيعملوا لهم activation. وحكينا كمان على المونوميريك بروتينز، monomeric G proteins. نكمل على regulation by changing the structure of enzymes. هلا حكايه الفوسفوريليشن حكينا عنه قبل شوي. هذا reversible. So it's a reversible covalent modification and it is really an efficient way of regulating enzyme activity. لأنها سريعة جدا. أنت بتعمل phosphorylation وعلى طول بتشيل الفوسفات، يعني it only takes few seconds, less than a second actually, إنك تعمل activation و inactivation للإنزيمز. لكن في عندنا كمان covalent فهذا reversible لأنه كان back and forth. لكن في عندنا كمان modification of enzyme structure that is irreversible. That is once the protein structure is modified, it cannot go back. That's it. It's modified and that's it. So, great example of proteins that are modified covalently and irreversibly is a group of enzymes known as zymogens. Zymogens are enzymes that require proteolytic cleavage to be activated. Okay? They require proteolytic digestion or proteolytic cleavage in order to be activated. So basically you have an enzyme, you have a zymogen that is not active. And it does have a peptide, part of it, that covers the active site. Okay? فعندهم الانزيم مثلا هيك وفي peptide جزء منه it covers the active site. So it prevents substrates from getting into the active site. What happens is that with these enzymes, with these zymogens, is that you have degradation, release, cleavage of this peptide. So now the active site is now open and it can, substrates can get into the active site. These are zymogens now.
العكس طيب انه كيف نعمل turn off activity تبع الانزيم؟
Okay. Notice that once this peptide is cleaved, it doesn't do any work anymore. It's cleaved. It doesn't go back and it doesn't get attached to the enzyme itself. So how can we inhibit this enzyme? There are inhibitors for other proteins or other molecules that bind to the enzyme, inhibiting it. That's one. Or two, the enzyme is degraded by other proteins. Okay? So, great example of zymogens are digestive enzymes, which are trypsin, chymotrypsin, elastase, pepsin in our stomach. These are all secreted as zymogens. Okay? So, and they're secreted by pancreatic cells. And this is a smart way. So the pancreas has these zymogens inside the cells. And whenever you eat something, a signal is transmitted from your mouth all the way to the pancreas, telling the pancreas, get ready, food is coming down. So it releases these enzymes as zymogens into the intestine. And inside the intestine, you have activation of these enzymes by proteolytic enzymes are ready for work, and once they are needed, they are immediately released. So they don't get produced, they do not get synthesized. Okay? And they stay in cells and tissues as zymogens. Why? Because these are digestive enzymes. They will digest anything. They are ready to degrade any protein in front of them, whether it is food or tissue itself. So if you have hyperactivation of these zymogens, of these digestive enzymes, you would get pathological conditions whereby the tissue is totally degraded, and it doesn't. And tissues are degraded like collagen, elastin, and all of these extracellular matrix proteins. Okay, so this is a smart way of regulating enzyme activity. I'm going to stop here. Oh, no,